So hopefully by now that you've done your own summary of your results and you've attempted all of the questions within a 60-minute time frame. So I just want to reiterate the results before we go through the discussion questions. So we had 4 different pHs, okay, and everything else remained the same. So at times you might get given a experimental design question, and it's really important that you know, and I'm getting off track here, you know what ditches stands for, okay? So you need to be able to identify the dependent variable, which is essentially the results. You need to be able to identify the independent variable, which is what is being tested or changed, and there's only ever one of them. If there's more than one within the one experiment or test, then it's unfair, okay? And the results will not be reliable. So these are the two main ones that you need to know about. When you're designing an experiment, you need to keep in mind the time period, so whether it's going to be 5 minutes, whether it's going to be a day, whether it's going to be a week, whether it's going to be a year. So you need to make sure that you're talking about that. Sometimes they give that a mark. I just added in just in case. C stands for, and I'm going to change the colours for these ones, controlled variables, so things that are kept the same. So when I was discussing the pH, we would say that the pH, because that's changed, that is the independent variable. Okay, this reaction chart that we're using to measure the results in is our excuse me, dependent variable. Okay, and the fact that we have the same amount of solution, the same time frame that it's in there, the same equipment that we're using, that's all controlled variables. Okay, now we have something also that starts with C called a control group. Now a lot of students get this confused. They think that controlled variables and control group is exactly the same. What a control group is a test that doesn't have an independent variable. So it will be whatever the normal situation is. All right. In the stem of the question, if you go back to that, it says that the hang on, I'll just find it and read it for you so you can see it. It says the optimum range of the phosphate that we'll be using in this experiment has a pH of 5. So that generally means that that's where it will be found in its normal environment. Okay, so a controlled variable is in everything that we do, so they're things that we keep the same in order for it to be a fair test. A controlled group is a type of test that we do within an experiment that does still have all the same controlled variables that we see in all the other experimental tests. However, it doesn't have an independent variable. Okay, so we wouldn't be, in this case, we wouldn't be exposing the enzyme to a pH that it doesn't normally, it's not normally subjective to. Okay. All right. Sometimes when you are asked to design an experiment, you need to be specific to your hypothesis. So what hypothesis you are you would be creating from your experiment, or what results you think that you might see from that hypothesis. And then last but not least, it's very important, so I'm going to put this in a different color just so it stands out. It's important that you have an accurate sample size, all right, or that you've repeated it, so repeated measures. I don't know if that's going to fit in. Repeated measures. It's not. So you need to have all of these things when you do come across a question that asks you to design an experiment. Now, we were really hoping for one last year, and we were lucky enough to get one, so let's hope for it again this year. It was worth 3 marks, and they needed to pretty much give all of this information here in order to make sure that they obtained those. Okay, so make sure that you're using ditches. It's a very important acronym. Okay, so I'm just gonna get rid of all of that so that we can now look at the results. Okay, beautiful. All right. So the results here, I would like I've said in the previous video, I want you to make a summary of what you see in the results. Okay, so this is the chart here that tells us the degree in which the reactions occurred. All right, so anything that's really pink, so I would say that really pink starts from about here. So anything from about a 7 to a 10, because that's the highest that we've got on the scale, they're at optimum. It's really working. Okay, so if we have a look at our results, is there anything that we can see that's at optimal range of pH? All right, so we've got our 9, and that sits about here. Okay, I'll try and color code these. We've got our 6. Okay, it's still fairly high up. It's not as a defined pink as the 9. Okay, so we'd say here that the enzyme hasn't denatured, so no denaturation. But we think that this one is more than likely it's optimal because it's working. It's the most effective at that. And remember that where it's most effective is because the conditions that it's in, the particles are going to collide more frequently. It's also going to have a lower activation energy that's required at that pH or at that temperature. Okay. Now, if we have a look at the other ones that we're seeing, we've got a 1. All right, and that comes all the way down here on the scale, so that's quite low. Okay, so I'm almost thinking that we've gone out of the optimal range and we're almost at the state where the enzyme is denaturing. Okay, and then the same here when we look at the 2. Okay, so if we were to be asked about enzymes and whether it was denaturing, we could discuss pH 3 and pH 9. Okay, now when we discuss temperature, so we'll just put that over there for a side. If we look at temperature and enzymes, and if you've looked at your cover page, you know that that's what it's on. Okay, there's a few important terms that you need to know. So you have your critical temperature, and you have your optimal temperatures. Okay, your optimal, all right, is obviously where the enzyme is going to work its best. Happy days. Okay, because we've decreased the activation energy, and we've increased the rate of collision. Okay, but when we get to our critical temperature, we've gone above the optimal and we started to denature. That's the point where denaturation occurs. And when we talk about denaturing, and I'm always running out of room, that's when we start to see the bonds. They start to break, and that's where the active site starts to be affected. And if the active site is affected, then it can't bind to the substrate. Ooh, sorry. Can't bind to the substrate, which therefore it can't create product. Okay, and that's when denaturing starts to occur. All right, so it's a little bit different with pH. You can denature it when you go above or below the optimal. Okay, whereas with temperature, when you go below, when you do below, the reaction slows down but it doesn't denature. You can heat it back up and it will still work. Okay, the only time that you will denature an enzyme when you're exposing it to different temperatures is when it's above. All right, when it's below, it will slow the reaction right down, but then you can heat it back up. As long as you don't heat it back up past that critical temperature, then you're okay. That's why they've, you guys know Walt Disney? That's why they've actually frozen his body, because they don't have the technology now, but he's on ice because his enzymes may still be working if they find the technology to I don't know, defrost him, I guess, and yeah, so to defrost him, because his enzymes aren't going to denature if they're frozen. There we go. Okay, beautiful. So now that we've got that out of the way, we can see that if we have a look back at the pH, so that pH of 3 and pH of 9 is going to below and above the optimal range for that enzyme. So then the enzyme has started to denature. When it does denature, remember that it's affecting the active site, and if it affects the active site, which is the 3D shape, because it's the tertiary level, then we can't bind to the substrate and we can't produce the product. Okay, and that is when we say it's denatured. Once it's denatured, it's broken, it's gone, we can't fix it. Okay, so it's really important that you know that you can't fix it. Okay, it's broken, it's done. Okay, awesome. All right, why are the enzymes extracted from the bean shoot named phosphatase? Okay, now when you're answering questions, to give the most specific answers, all right, I would like you to provide like a blanket definition and then relate it back to the experiment. If you just relate it purely to the experiment and don't provide that blanket definition, you are not going to get the marks. All right, if you just give a blanket definition, depending on the examiner or the year of the exam, to be honest, you might get the mark. So go with that. If you get a question and you're not too sure and you just put down a definition, that might help you. But generally go with definition and then example from the scenario that you've been given. So we know that phosphatase is an enzyme. So I'm just going to try and dot point this out for you. All right. Obviously, it's going to be too hard for me to write on my tablet as we're doing this. So this is a organic catalyst, which is an enzyme. Okay. Organic catalyst, and it's an enzyme. We already know that all enzymes end in ASA, or majority of them do. Okay, and then what's its role that it does? It binds to phosphate. Okay, which is exactly how it's got its name. All right, so it binds with a phosphate. It's an enzyme. It helps to speed up reactions. All right. When you do talk about organic catalysts and enzyme, there are other different types of organic catalysts. The difference between all organic catalysts and an enzyme is that it's made of protein. So I might even put that in there as well. All right. So it's an organic catalyst that's made of protein that speeds up reactions and is not changed. All right. Speeds up reaction, and it's unchanged. So pretty much what this question was asking you is, what is the role of an enzyme? Okay, and then you could talk about why it is called that, is because its substrate is phosphate. I hope that makes sense. Okay, I might even change colours for when we go from different questions, just to try and make our lives a little bit easier. Okay. Yeah, because we're going to need some space for this one. Chemical reactions involving synthesis, and metabolism of glucose. So remember how I was saying to you in, I think it was the topic test 1 video, that glucose is extremely important. Okay, so glucose, when we think about glucose, you've got to think about where it's produced, which is in photosynthesis. Okay, and then it's used in cellular respiration. So remember, all organisms need to go through cellular respiration. Autotrophs will go through photosynthesis or chemosynthesis in order to make their own source of energy, in this case glucose, but they all need to break it down further, which is cellular respiration. I'm hoping by this time of the year you know the balanced equation for both of these reactions, and that you know that there are 2 different types of the 2 stages of photosynthesis, which you have your light dependent and your light dependent and light independent. Okay. And then that you have 3 stages also in cellular respiration. So you have glycolysis. Oh, move out. Gosh, I don't even know how any of us could read that. Glycolysis, Krebs, and then the electron transport chain. So you'll definitely need to know those different sections of those equations or reactions. Very important. Okay, so when we refer back to the question and it's talking about the synthesis, so that's photosynthesis, hence its name, and then cellular respiration is metabolism. All right, which in this case is referring to the breaking down of glucose. All right. What role would protease have in aiding these chemical reactions? Okay, so remember when we first looked at some of the question, we said that we thought of phosphate as being in ATP, in nucleotides, and in your phospholipids. In this case, we're talking about photosynthesis and cellular respiration, so I think that they are definitely talking about ATP. Okay, now we're building, in photosynthesis, we are building up, we're creating the glucose, so we're going to need energy. All right, whereas in cellular respiration, we're breaking it down, so we're releasing energy. Okay, so the way in which this cycles within a cell, okay, is we have ATP, all right, that's carrying energy. When that's broken down, it goes into ADP plus PI, which gives off energy. All right, this enzyme would help with the cycling of this. Okay, so it might join to the ATP, help to clinch off one of the phosphates, which then releases the energy. Okay, and remember, enzymes can work in both directions, so it could also add that phosphate group back on and take in some energy to give us the ATP. Okay, so it's really important that you understand that this enzyme would be important for binding with the free floating phosphates that are in the cytoplasm, as well as the ATP molecules to clench them off. Okay, so it's definitely the cycling of the ATP that provides the energy and takes up the energy for these important reactions. Okay, I think I've talked enough about that one. All right, so question 3, let's zoom in and have a look at that. All right, so question 3. What would you conclude to be the optimal pH based on your results? What is meant by the term optimal in reference to this experiment? So this is great because you've already done your summary. Okay, so if you go back and you have a look at that, you can see that the pH where the optimal range is would be somewhere between, based on our results, pH of 5 to 7. Now, most of the time they don't really like you to give a range. They want you to be specific. So they haven't said the optimal range of pH, they've said the optimal pH based on your results. And because we've got a 9 on the ranking chart or the rating chart versus a 6 for 7, I'm going to say that the pH for this is pH 5, which is the optimum because that was 9. Okay. And that's what they want you to do. So you could do some research and that might not be the optimal pH, but based on what you saw, that's correct. You could then, if you knew for sure that it wasn't that, and for example, it could have been pH 9 and we got the wrong results, then you could say, in our results, we saw that the optimal range was pH 5, but then, however, we should have got a pH of 9. Because it lives in this sort of environment, or something along the lines of that. Okay, so then second part of the question, remember this is only worth 1 mark, but it's asking you 2 things. If you don't include both of them, you are not going to get the mark. All right, so this is where I talk about reading the questions really important. Okay, so what is meant by the term optimal in reference to this experiment? So again, make sure you're talking about a blanket definition and then talk about that. All right, so the optimal refers to like I was saying before, the greater number of collisions with enzymes and substrate, and therefore has a lower activation energy. Okay, so the greatest reaction is achieved. So I'll just jot them down. So the lowest activation energy, increased collisions, right, it's the most effective. Okay, and so then you would just say that this was occurring at pH 5 in this practical. Okay, all right, question 4, I might just move this up a little bit. Change colour. Which buffer showed evidence of denaturing of enzymes in this experiment? Upon close examination of the denaturing enzyme, what changes might be observed? So again, you need to really talk about what you saw in your results, what you might have needed to have seen or what you expected to see, and then talk about the denaturation of an enzyme. Okay, so you would say that at pH, what was it, pH 3 and pH 9, the lowest reaction that we saw was at pH 9, so I'd probably talk about that. Again, we don't want to range, okay, but I'll write it down anyway. pH 3 or pH 9, we had a reduced activity. Okay, so we would say that denaturation maybe started to have occurred. Okay, but again, it could have been just slower and it could have been the time period. So you don't need to talk about that, but take that into consideration. And when you're talking about denaturation, this is so important. Okay, you need to know that whatever exposure that the enzyme has had, that's started to affect the peptide bonds. All right, which then starts to affect its structure, which therefore it changes shape, changes the active site's shape. This is really important, okay? And therefore cannot bind to substrate, which means no product. Sad face. Okay, really important that you were talking about that. All right, so the active site of the enzyme would have changed shape, and this means that the enzyme is no longer able to effectively bind with the substrate. Okay, therefore no product. All right, I think that's fairly easy to remember. So just please make sure that you are very specific to that. All right, because a lot of students miss out those details, and I think that it's not too hard of a concept to get your head around. Okay, beautiful. All right, I love this diagram. I think it's really nice. No, what color should we go for? Green. Okay, so again, it's a diagram. I'd probably label it before I did it. So have a look at C. So that just looks like it's an enzyme, maybe. I reckon that. Or, oh no, if we have a look. So we go from C and we follow it goes there, and then it changes. So C cannot be the enzyme because that has changed. So that means that that must have been the substrate. Okay. So substrate, and then if you have a look up here, it says in relation to the experiment as well. Okay, so then you could say that that was the PPP. All right, or even if you said phosphate, that would be okay as well. All right, E, go to that because it's okay. So that's obviously the product, which is your free phosphate and that phenylalanine. So I'm not going to have a space to write that. Okay. So F, that's obviously still the same. That hasn't changed from here to here to here. So we'd say that that's definitely the enzyme. Okay, then we can work backwards. So we've done C, A. So that's pointing to the enzyme, and that would be pointing to the the active site. Okay. If that in fact was pointing, hang on, let me write that. If that in fact was pointing in this direction towards the substrate, then we would say that that would be the binding site. Okay, so that's important to know. So the active site binds with the receptor at its binding site. Okay, now when the substrate and the enzyme are together, we call that a substrate, enzyme substrate complex. I did it again, didn't I? Enzyme substrate complex. Okay, and then again we've already said that B must be the enzyme. Okay, I would know that diagram. Okay, let's go up. Question 6. Describe the region. Oh, we need to see that diagram. Describe the region labelled A above and explain its role. All right. So A, active site. Okay, so the active site refers to a specific region on the enzyme where the substrate can bind to it. Okay, and it needs to be specific to shape. So the things that I would include, obviously what it's called, so it's an active site, specific 3D shape, and it can bind to binding site on reactants. And if you really want to, allows for product. To be formed. Okay, so just like in question 4, it's pretty much exactly the same thing. We're talking about its structure, we're talking about its function, we're talking about the active site, we're talking about shape, we're talking about the binding site, we're talking about reactants, and we're talking about product. Okay, so really main concept that you need to get your head around and you need to talk about. Okay, 7. The reaction seen in this experiment is summarized in the following diagram. Okay, so this is exactly the same diagram that we saw in the stem of the question. All right, explain how the induced fit model would facilitate this particular reaction. In your answer, include in your answer how this model would differ from the lock and key model. So again, like I was saying at the start, you didn't learn about this reaction specifically, but we have talked about induced fit and we have talked about the lock and key. Okay, so the lock and key, I want you to think about it literally like a lock and key. The key needs to be a specific rigid shape to get into the lock and to turn it. Okay, whereas when you go up and you think of the induced fit, like when you go up and you give somebody a handshake, okay, your hand molds around their hand. Okay, or even when you go up and you give somebody a hug. All right, you're going up, and then because you're binding with them, that's creating them, in molecular terms, then they're changing shape and molding to your body. Okay, so that's the analogy I've got for that. So the lock and key, and I'll just write it here, is a very rigid fit. Okay, whereas here it's a bit more flexible. And the place that we're talking about is the active site. Okay, so when you were talking about the induced fit, it's flexible, and when they bind together, it induces a change in shape. So the molecules are almost hugging. So when the reaction is taking place, they're still quite close together, but they're not as... when they come together, that induces the change in shape for them to be so close. Okay, and you'll see that pattern reoccurring a lot. So whenever molecules do come into contact with each other, just like we were looking at the receptors in the topic test video, so specifically the G protein that we didn't talk too much about, but we will in the future. When that binds with a receptor, that changes a lot of the one of the shapes of the G protein complex down the bottom, which causes the changes and for that stimulus to be passed on to the inside of the cell. So it's really important that you understand about this induced fit and the induces in changing its shape. Okay, so please make sure that when it asks you to compare, that you've got words such as like whereas, therefore, or compared to. Okay, just to check your own work. So if you're looking for these words when you're just going through reading time, you maybe got like 5 minutes left before you got to put your pens down, double check to see whether you've got those words there so that you are making a direct comparison. I know this is a little bit off topic, but please do not start your responses with this durated word, because it's not that you're going to lose marks, but you are limiting your responses by starting with the word because. Okay, so please keep that in mind. All right. So question 8 is a graph. I'm sure all of you know by now that I love graphs. I love tables. I love visual representations of things. So if you haven't found your love for graphs, I suggest that you do start to find that now, so that by the time it comes to the exam, when you see a graph there, or in any topic tests or SACs that we do throughout the year, you get excited. Okay, get that adrenaline pumping. So graphs you will see for any type of reaction. So whenever we're looking just at a blanket enzyme, just like we are now, or when we're having a look at the relationship between photosynthesis and cellular respiration, or even those two equations separately. Okay, you'll need to be able to annotate your graph. So have a look at the axes, have a look at the reaction. You might need to, excuse me, complete the graph. You might need to annotate the graph. You might need to respond to the graph. So these are the sorts of things that you need to be able to do. All right. So let's zoom in and read what they're asking us to do. So if the incubation of this experiment was not stopped, so meaning that we didn't add the sodium carbonate, so remember that that provided us with that basic environment, which did two things. It stopped the environment, and then it also gave the phenolphthalein an environment where it was able to show its pink colour. Okay, because remember before that it was colourless. All right, so if we didn't add that and we didn't stop the reaction, and then the reaction did continue to occur until no phenolphthalein phosphate remained. Okay, so there was no PPP. So that just kept going until it broke down into phenolphthalein and phosphate. Okay, the amount of free phosphates produced could be measured and reconnected throughout the experiment. Draw a graph to show the change in phosphate concentration in the test tube. Okay, so change in phosphate concentration. So remember, if we're looking at this as a reaction, this here is the reactant, and then these are the products. So the amount of phosphate, this is the amount of product. All right, and it's over a time period. Remember, 0 generally will always go here. So when we're looking at product, sometimes you could even infer about the rate of reaction. Okay, so how fast it's occurring. So you would say that, so here we've got, I'll change colour. So here, down at 0, that this is where the reaction first starts. So we start here, where all we have in the test tube is the PPP, okay? Then once the enzyme starts to work, okay, then we gradually get more and more and more and more phosphate, because remember that's what we're looking at. And then we reach this certain point, okay, where the time's increasing still. However, there's no increase in the amount of phosphate. All right, so we're not getting any more phosphate. The reason being is that we have used up all of our PPP, all of our reactant. So if we don't have any more reactant, the enzyme has nothing else to work on, so therefore we cannot make any more phosphate. Okay, so the limiting factor here, the thing that's stopping the reaction, I'll just write it here, limiting factor, because there is always at least one limiting factor that will limit the reaction rate, and that's the amount of reactant that we have here. Okay, so this is the sort of thing that you're going to need to be able to do. You're going to be able to need to write and annotate on the graph. Now I'm just going to come over to the side here because there's a few other things that I want to go through with you in terms of graphs, because they will come up, and I know some of you aren't extremely keen on them. So let's go with a purple colour. Yeah, that's one. Okay, so I'm just going to write the reaction at the top, the one that we've seen in this practical. So we've got our PPP, enzyme comes along, breaks that down into phenolphthalein and phosphate. So remember, this is the reactant. These are the products. And then this here is representing our enzyme. It's done it to me again, isn't it? I don't know what happens. so we're just going to come over here. I'm just going to write the reaction that we've seen in our practical. So we've got our PPP, which ends broken down by an enzyme to phenolphthalein plus phosphate. Okay, so this here, remember, is our reactant, or because we're talking about enzymes, it's our substrate. Over here is our product, so what the enzyme's creating, and then our enzyme is represented here by our arrow, meaning that a reaction has occurred. Okay, so the different things that they might throw at you is they might ask you about concentration of the substrate. And what's that? All that means is it's the amount of substrate that you have. Okay, they might also ask you about the concentration of an enzyme. Okay, and then different things that they might ask you about the product, or to infer about the product, is the rate of reaction. Okay, and that might be against they might put that against time. So just like we saw in the graph over there. Okay, they might put that against substrate concentration. Okay, they might put it against enzyme concentration. Or they might even put it against, you would have seen these in your weekly tests, they might put it against temperature, and they might put it against pH. Okay, now these sorts of things, when they're putting them against temperature, is that they will be looking and testing your understanding of denaturation. Okay, so remember, so I'll just do an example for you for these ones here of temperature. And so they might have the rate of the reaction over here. So remember, that's the amount of product that's being produced. That's what all that's asking you. Okay, 0 will be here. And let's put it against temperature. All right, and this is just going to be rough, and it's not accurate. I'm not using tails. And that's 30. And let's just say that this is an enzyme that we find in humans. Okay, so if it's at 0, chances are that it's the rate of reaction, so the activity of the enzyme. I'll just write that up here too. Activity will be less, and it will increase because our bodies are around 30 degrees, so it'll increase there. But then as soon as it starts to get past that 30 degrees, the enzyme's not as keen on doing that. And then after a while, some of those the bonds within that enzyme might start to break, and therefore the reaction's decreasing until all of the enzyme is denatured because it's been exposed to higher temperatures. Okay? And some of the questions that you might get asked are things like, what's happened on this side of the graph? Okay, and for temperature, you need to know that this has just slowed down the reaction. It hasn't denatured. Okay, however, over here, this is denatured because it's been exposed to its critical temperature, and then it's not going to work again. If you heat these enzymes up here that have been exposed to cooler temperatures, if you expose them up into the optimal range, which is around 30-37, then it will start to work again. Okay, but once it reaches this critical temperature where it starts to denature, that's it. The enzymes are gone. Okay, so So that's really important to know. Whereas pH, it can denature at both extremes. Okay, however, its range for pH is probably a lot longer, a lot bigger than it is for temperature. Okay, so I just want to show you a few more examples because this will really come up a lot, and I think this is something that's really easy, but a lot of students get it confused. Okay, so whenever you do get a graph, please make sure you annotate it just like I've done there. So what I'm going to do is I'm going to draw a graph, and I'm going to put the rate of reaction over here, and then I'm going to put substrate concentration down here. So remember, substrate concentration is with this experiment, we're looking at our ppp. Okay, so again, 0 is always here. All right, so the amount of ppp that we have, the reaction, because the enzymes, it's going to be moving across, okay, and there's going to be less and less concentration. Hang on, what it's asking you is if the substrate concentration is being increased and increased and increased, okay, and this is the amount of product that's being produced. All right, how fast that's occurring. So you can increase that substrate concentration and increase it and increase it and increase it. However, if you don't, if you keep everything the same but only increase your substrate concentration, you're going to do something that we call saturate the enzymes. Okay, and it's going to get to a point where all the enzymes are working at their fastest. So all of them have got a reactant, then they're breaking it down, then having a product. They're getting another reactant, they're binding with that, and then they're producing a product. So I keep calling it reactant substrate, and they're just working at 110%. Okay, and we keep increasing the concentration of the substrate, but the enzymes aren't going to work any faster because they are already at their maximum. Okay, so this is the limiting factor that we can see on the graph. So the limiting factor would be the enzyme concentration. Okay, because that's what's preventing the reaction from increasing. Okay, if, I might just get rid of this. If we were to just say that you had to put a graph, all right, you had to write in onto the graph certain things. So I'm just going to fix this up for us. All right, and I'll put time down here. So we've got time here. All right, so let's just say this is our standard graph that we've just drawn for question 8. Okay, but then what happens if the question says to you, in green, I would like you to show what would happen if you had doubled the amount, double the amount of enzymes. Okay, so if you double the amount of enzymes, is that going to change the amount of product that you have? No. Okay, so the only way to increase the amount of product would be to increase the amount of substrate. All right, but if you increase the amount of enzymes, that work that you get done would happen in less time. Okay, so instead of the reaction finishing up here, it might finish a lot sooner. All right, we'll get to its peak. So it might do something like this. Okay, so it's got to its limiting factor a lot sooner because it has a lot more enzymes to get the job done. If I doubled the substrate concentration, Okay, so this is the amount of product that I was producing. Now this isn't going to be accurate because I haven't left myself enough space unless I do this. That's better. So this is the amount of product that's been produced. So let's just say let's just give it a number. So that's 5, okay? So that's the amount of product that I have. If I double the amount of substrate, that means that I'm going to have double the amount of product. All right, so I haven't changed the enzyme concentration, so it might still happen at the same time. All right, but in fact, I'm going to have double the amount of product. Products over here. So if I go over here, this would have been a 5 over here, and now we're at a 10 because I had double the amount of substrate. So therefore, I can have double the product at the end of end of the reaction. Okay, so it's really important that you are reading what's on your axes because that can dramatically change what it is. So I would make sure that you're having a look at these sorts of variables. When you come across any sort of graph, when it's your weekly questions or a practice SAC or a practice exam, go through and with a graph that you get and change those things around and play with it and see if you know what to do when you do change those variables. Okay? Question 9. Explain what is meant by activation energy. This sounds like something from Power Rangers, but I don't know if you guys even know what Power Rangers is. So activation energy, okay, is in a biological reaction. That's the energy that is required to start any reaction, okay? The reason why enzymes are so necessary is because it lowers that activation energy. So all they're asking you here for is it's the energy that's required to start a reaction. Required to start a reaction. Okay, beautiful. Number 10. So, an enzyme can be said to catalyze a reaction in both directions. All right? Because think about it. It binds with, let's just say we've got 2 substrate molecules. Okay? It binds with both of them and then makes 1. All right? Either way, it's still able to bind to both shapes. All right? So it can go in either direction. That is, the phosphate could also, given the right conditions, combine phenylalanine and phosphate together again. Okay? Explain how the reactions in this experiment in its forward and reverse directions could be explained as catabolic and anabolic. All right? In your answer, also explain the energy required of each reaction. Okay, so they're asking us to show, and you can use diagrams in this sort of scenario as long as they're annotated, okay? So we need to talk about the forward and the reverse directions, all right? Catabolic, anabolic, and energy if it's required or if it's an output. So what I'm going to do is I'm going to do it over here. Okay, and I'm going to keep a little dot points of what we need. So we need to have forward reaction, we need to have a backwards reaction. And we need to talk about energy, and catabolic, and anabolic. Okay, so the forward direction would be what we've seen, PPP, all right, and then we have our phosphate plus our phenylalanine. Okay, So that means that the backwards reaction would be your phosphate plus your phenylalanine, which reforms your PPP. Okay? So in this case, what we're doing, so we'll have a look at anabolic and catabolic to start with. All right? So we're going from PPP and then we're breaking it down. This is breaking down. So it must be releasing energy. Releasing energy. So for releasing energy, it's catabolic. Because we're breaking it down. And because we're breaking it down, it's breaking down those bonds, which was releasing energy. Okay? If it's releasing energy, okay, then we call it exergonic. Exer, it's exiting. Gonic refers to the energy. Okay. Now we'll have a look at the backwards reaction. Okay, so instead of going from the PPP to the phenylalanine and your phosphate group, you're now going from your phosphate group and your phenylalanine, and you're building it up to a PPP. So you're building up, just like estrogen. You want to get bigger. So this is an anabolic reaction, and you require energy. Takes a lot of energy to build all those muscles. Okay, requires energy, so you're going to need to take in energy. Okay, so it's going to be an endergonic reaction. So the words that they're after here, what they want you to have, is this forward reaction, this backwards reaction, and then they'd like you to describe each reaction here as either catabolic and anabolic, as we've done, and then exergonic or endergonic. We've done that here. Question 11. Temperature is an important controlled feature in this experiment. So remember, this comes back to your understanding of ditches. So controlled feature. Explain how the results of this experiment could be affected if one of the test tubes was put into the refrigerator. So remember, a refrigerator is a colder temperature. Okay, instead of being incubated at 30 degrees, because remember that was again controlled feature, so all of them were at 30 degrees. So it's asking you, if you were to put them into a colder environment, what's going to happen to it? Now I want you to start thinking about the enzyme activity when you read this question. When you think of temperature, I want you to think automatically to think of enzymes, and I want you to think about what type of response that's having on its activity. So if it's going to be in a cold environment, it's going to induce a reduced rate of reaction. So I'm just going to write some dot points here. Reduced rate of reaction. So therefore, the reason why we're having a reduced rate of reaction is because there is a decrease of collisions. So the particles aren't moving anywhere near as fast, so therefore the substrate is less likely, or it's going to take longer to find the enzyme for it to bind with it. Okay, so therefore if the collisions between the enzyme and the substrate are reduced, okay, therefore that's going to slow the reaction down, and we'll see that by either less product within the same time, in time frame, okay, so I think that one's fairly easy. Beautiful. Question 12. So a student hypothesized that phosphatase taken from the roots of the bean plant would have a different optimal temperature from that plant above the ground. Okay, so we can see here in the diagram, so this is where they differentiate. So the one below the in the soil is 15 degrees, or they're above, where it's exposed to the sun, is 25 degrees. Okay, do you agree or disagree? Explain reasons for your choice. So this is worth only 1 mark, and you need to agree or disagree. So you need to state whether you're going to agree or disagree, and then explain. Okay, so when it says explain, I would always go back to your scientific understanding. Okay, and I just want to compare that to if you get given a graph and they say, so you might be asked to explain or I've lost my train of thought now. I'm just going to go through explain. So explain is your scientific understanding, whereas the other one, the keyword I can't think of right at the moment, it's going to just ask you to describe. Oh, that's right. Describe. Describe is just going to ask you to tell them about the trends. If you're asked to explain something, you need to talk about what's happening in terms of scientific understanding. So in this case, you're going to talk about enzymes. Okay. All right, so use your scientific understanding for this one. So you could have 2 options here as long as you are able to justify a response using this understanding of enzymes. Okay, so you might say, let's go first of all with agree. So you might agree that they have different, just go back up so you can see it. So you might agree that they have different optimal temperatures. Okay, so because the roots grow at a soil of 15 degrees, it would be expected that their optimal temperature would be around that, and then the same with the above ground, so that's exposed most of the time to 25 degrees. So you might say with agree, so they have different environments. So it makes sense for them to have different optimal temperatures due to that. Okay. Sorry, I'm just going to dot point it because you know I run out of room. Okay, but then you could also go with disagree. All right, again, as long as you're justifying yourself with your scientific understanding of enzymes. So you could say that it would be expected that they'd have the same optimal range because they are in the same plant, and so they'd need to be the same enzyme because they're made by the same organism. Okay. So you'd say it'd be expected the enzyme is the same because it's the same plant. All right. Same plant. All right. So as long as you're justifying that, in this case, you would get the answer right. As long as, again, you're coming back to your scientific understanding. All right, I can't stress that enough. Beautiful. Now, this question here I'm not going to spend too long on because rational drug design is actually not in the course anymore. So this is an older SAC that we're going through. So pretty much up here it just says to you that medical research are starting to investigate the use of the enzyme in treating bacterial diseases. So it does alter some of its what should we say, abilities. Okay, so some of the things, so motility, so its ability to move, uptake of nutrition, metabolic regulation, cell division, so a lot of its processes, okay, that they're looking at using this enzyme to treat bacterial diseases because it affects a lot of its essential processes, basically. Okay, so the first question, 13 there, is what is rational drug design? Again, this isn't on the course, but I'm just going to go through it anyway because I think it's interesting to know about. So rational drug design involves the analysis of a disease to determine a structure of the disease, and then a drug is then designed to block it. So pretty much we're looking at inhibitors. Okay. So yeah, so they're designing a drug that's specific to that active site, which will bind with that and prevent things from occurring. So in this case, we would have our enzyme, okay, and we know that that binds to phosphate and let's just say phosphate. Yeah, we'll just go with that because that's how we always draw it. So therefore, they might find a drug or develop a drug that is exactly the same shape as that. So then that competes for the active site. So when they would add this drug into it, this would bind with the enzyme, which would then affect the processes. Okay, so that's essentially what it is. Okay. All right. Lucky last question. So, why would medical researchers be interested in the effect of using an inhibitor to block or inhibit phosphatase using such as an ATPase in bacteria? Okay, so ATPase, you guessed it, it helps in making ATP and it's an enzyme. Okay, so the reason for being that, and hopefully you've got the big picture, so it would slow down chemical reactions in the bacteria because it would be reducing the amount of energy that it has. So reduce its energy. In this case, it's ATP. Okay, which would then affect its processes. All right, fairly straightforward, but I wouldn't worry too much about these two questions. I doubt that you will get them. It is important that you do know about inhibitors, so I might just come over here and briefly, get rid of that, briefly go through them. Inhibitors. Ooh. So I'm just going to use shorthand, but you've got competitive and you have non-competitive. Okay, on an enzyme, I'll just draw the one that's in our classroom. That looks a bit funny, doesn't it? So this is your enzyme. And this is the active site. And this is the site here that we call allosteric site. Okay, the products bind in here, the reactants. Reactants bind here and therefore create your product. So that's the active site. Competitive inhibitors will bind into the active site. Okay, if you increase the concentration of reactants, okay, this competitive inhibitor will feel threatened, intimidated, just like a dolphin and a shark. So if there's one shark and one dolphin and the dolphin's picking on it, so the shark would be the inhibitor. Okay, and the reactants would be the dolphin. The more dolphins that you get and they come along, all right, that shark will actually then move. All right, so then that will expose the active site again, and then the reactants can then move in and then form your products. Okay, so you're able to move that inhibitor. All right, non-competitive inhibitors. Okay, this binds not with the active site, this site at the back, the allosteric site. Okay, it doesn't matter how much we increase the concentration of the reactants or how many dolphins we have, it doesn't matter. That's not going to budge. Okay, and that's when we start to look at toxins that affect our body. We can, sometimes we do have natural occurring non-competitive inhibitors within our body, so that to assist in regulating certain things. But in order to get the non-competitive inhibitor out, generally there will be an enzyme that will then bind to that and break it down. Okay, I haven't talked about the most important reason why this non-competitive inhibitor works. I know you're really excited to find out. So what happens is when it binds in here, okay, it actually changes the shape of the active site. Okay, and again, I know that you're very aware of this, but if you change the active site, that's not going to be able to bind with these reactants, and we are not going to be able to form products. Okay, so that's when it's a toxin. Sometimes there's toxins that bind with enzymes within the mitochondria, and therefore that reduces the amount of ATP that we can produce, which can be extremely deadly because we need ATP for so many things in our body. Okay, so we need to, that's important. And then regulating, so we might want to create molecule A, but then not all the time. So that's why we have a natural occurring non-competitive inhibitor, which would prevent us from making molecule A. But then when we need more of molecule A, a particular enzyme might come along, bind with that, break it down, and then the active site will go back to normal. Okay, and then more reactants can go in to the active site, bind with that, and produce the product. Okay, so hopefully that helps.